Welcome everyone to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Refining Toxicity Testing Through Advances in Human iPSC Technology. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The rapid and efficient assessment of drug toxicity has been a constant challenge for drug development researchers. A leading cause of failure for drug compounds has been the lack of appropriate in vitro models that fail to recapitulate important aspects of biology, as well as identify adverse drug interactions. Consequently, novel approaches are critically needed to improve predictive toxicology. Rapid advances in the life sciences field have afforded investigators the ability to utilize advanced cell-based assays for surveying toxicological effects of various drug compounds. However, these assays fundamentally rely on the appropriate cell source for proper validation. Many of the properties of human-induced pluripotent stem cells, or iPSCs, make them a favorable cell source for toxicity assessment. Let's meet our panel for this GEN webinar, who will tell us how advances in human iPSC technology can enable early detection and avoidance of neuro, cardio, and hepatotoxicity during drug development. Our first speaker today is Brad Swanson, Vice President of Cell Biology R&D at Cellular Dynamics International. Dr. Swanson will explain the advantages of studying neurotoxicity while using the iCell gluconeurons. Dr. Swanson will be followed by Andrew Bruning-Wright, Principal Scientist at Charles Rivers Laboratories. Dr. Wright will discuss data from his studies using iCell cardiomyocytes that help validate this line as an appropriate tool for cardiac drug toxicity testing. Our final speaker will be William Proctor, Investigative Toxicologist in the Department of Safety Assessment at Genentech. Dr. Proctor will describe his work on preclinical toxicity testing using iCell hepatocytes. Before our speakers get started, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We'll try and answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, so without any further ado, let's get our webinar underway. Brad, take it away.
Thank you. The title of my talk today is iCell gluconeurons, a new model system for studying neurotoxicity in human cells. My presentation today will be in two parts. The first part will be a brief overview of CDI, the company, and our products and the services we provide, and the majority of the time I'll spend after that discussing a new product that we have recently introduced to the market, iCell gluconeurons. And in particular, I'll be focusing on two aspects or two use cases of iCell gluconeurons, using them to study excitotoxicity, and also looking at studying these cells using the multi-electrode array to study synaptic activity of human cortical neurons. First, a few slides about CDI, the company. We are the world's largest producer of human iPS cells and iPS cell-derived tissue cells. Our infrastructure, we have approximately 170 people working at CDI, and we have a tremendous amount of human stem cell experience resident at CDI. We've been developing iPSC-derived human cells for the research market from day one of iPSC's existence. We're headquartered in Madison, Wisconsin, and we have a large number of patents owned or in-licensed to CDI, and the reason that we have these is to provide clear freedom to operate for the customers that are using our products. We have several areas of key competencies that we think are very important to provide the highest quality products to our customers, being able to reprogram iPSCs, being able to engineer the genome of iPSCs, as well as being able to differentiate iPSCs in a number of different cell types, and to understand how to use those cells to help answer important biological questions. Currently today we have a large and continuing to be growing portfolio of iCell products. We have listed on the slide here over 10 iCell products as well as our services for reprogramming cells into induced pluripotent stem cells. These products cover a wide range of biological cell types, including central nervous system neurons, a number of different cell types there, astrocytes, I-cell neurons, I-cell dopin neurons, and as well as the gluteneurons, which are not listed on the slide, which I'll be talking about later today. In addition, we have a number of other different cell types in the muscle lineages, such as skeletal myoblasts, cardiomyocytes, and our new cardiomyocyte squared product, and cardiac progenitor cells. Endothelial cells, macrophages, hematopoietic progenitor cells, hepatocytes, which we'll be talking about later today, are also included in our product portfolio. There are a couple of new cell types that aren't on the portfolio as well, and I'll be talking about gluteneurons, which is one of those today for the remainder of the talk. Our iCell catalog products are iPSC-derived cells derived from a single individual donor. We also offer a diversity and disease portfolio, which is where we can make custom background differentiated cells from basically any iPSC that we can obtain or our customers can obtain and present to us for differentiation. We also offer custom reprogramming from any individual, as well as custom genome engineering services as well. iPS cells provide a unique access to human biology and the ability to study that biology in the dish. We've demonstrated across many of our cell types the unique functionality that these cells provide when studying disease modeling or studying cytotoxicity or toxicity models across many different cell types, including our neuronal products, our cardiomyocytes, and the hepatocytes, which both will be covered in later talks today in this webinar. Another key aspect that comes along with our iCell products is not only do we provide these cells in the media, et cetera, but we also provide an extensive support system with the cells to help you use the cells and get the best results from your experiments. Our application support team has spent a significant amount of time and has partnered with a number of different platform providers of instrumentations or other assay readout technologies to make sure that when you get the cells and you use them, that if you want to use a particular platform or a particular assay system, that we can support you and get you to have results as quickly as possible and the highest quality as possible as well. For the remainder of the talk, I'm going to focus on a new product that we have in the market called iCell gluteneurons. These are human cortical neurons that are largely made up of an excitatory or glutamatergic neuron population. And this is a product that was actually based on market feedback from customers who had used our original iCell neuron product. iCell neurons are a product that have been widely used in the market, and however, they're largely made up of GABAergic or inhibitory neurons, which limits a number of the application cases for those cells, and there's a significant interest in having excitatory neurons to study for a number of different areas of biology, and that is the reason that we have developed this as a new product. These iCell gluteneurons are derived from iPSCs from normal healthy donors, and we think their usage will be targeted largely towards studying excitatory toxicity to model a number of different diseases, as well as studying electrophysiology to explore excitatory signaling. And more specifically, we think these will be a good model system for studying Caesargenic models. This slide demonstrates the general morphological properties of iCell gluteneurons once you thaw them out of cryopreservation and culture them for a couple of weeks. The photomicrograph shown here demonstrates iCell gluteneuron culture after 2 weeks of culture, and these cells were seeded at 80,000 cells per well in a 96-well plate, and then they were processed to be stained for calcium AM to demonstrate the neuronal processes present in the culture. And as you can see, within 2 weeks, actually even sooner than this, you see extensive neurite outgrowth from the cells, and the cells form a robust neuronal network within this period of time. Critical to understanding the inhibitory versus excitatory phenotype of iCell gluteneurons we've characterized their gene expression for a number of genes that are indicative of either excitatory or inhibitory neurons and compared them to our eye-cell neuron product, and this data is shown on this slide. Now, 3 lots of each cell type were analyzed for gene expression, and the gene expression was normalized to GAPDH. And the important take-home message from this slide is that the eye-cell neurons have a higher expression of GABAergic markers that are circled and labeled there in the GABAergic circle compared to the eye-cell glutoneurons. Conversely, our eye-cell glutoneurons have a higher expression of glutamatergic markers than eye-cell neurons, demonstrating that the eye-cell glutoneurons are enriched for glutamatergic excitatory phenotype neurons. To look at this more specifically, we undertook looking at gene expression on a single-cell analysis basis for our eye-cell glutoneurons. And this was done by actually sorting cells by flow cytometer into individual wells of a 96-well plate, and then processing these individual cells for a single-cell gene expression using a fluid ion biomark system. In this particular analysis, we had a panel of 48 genes that represented a number of different markers for different areas of central nervous system biology, excitatory and inhibitory neurons, as well as some regional markers. But we're focusing this slide mainly on the 2 sets of genes that are circled in the green boxes. Each point across the x-axis is an individual cell, and on the y-axis are the different genes that were analyzed for each cell. What this heat map shows is that for eye-cell glutoneurons, there's a majority of the cells that have that exhibit the glutamatergic gene expression fingerprint, shown in the box that's a little bit higher in the figure, and a minority of the cells, again, since these are glutamatergic enriched population, exhibit the GABAergic gene expression fingerprint, shown in the box in the bottom left hand of the panel. This again demonstrates that our eye-cell glutoneurons are enriched significantly for glutamatergic phenotype neurons. One of the main uses for eye-cell glutoneurons is to study glutamate toxicity. And this figure shows how you can actually kill eye-cell glutoneurons in a dose-dependent fashion when you treat them in culture with glutamic acid. And we've demonstrated 2 different ways you can read out this assay, either using cell tetraglow or detecting accumulation of lactate dehydrogenase in the cell culture medium. Now, to do this particular assay, the glutoneurons were plated in brain fizz medium in either 96-well or 384-well plates. Cells were cultured for 14 days and then were dosed with glutamic acid. They were allowed, the glutamic acid was applied to the cells for 2 days, and then we used the detection reagents, either cell tetraglow or LDH, to determine cell viability and cell death. And you can see there that they have the same basic EC50 for response to glutamic acid with either the cell tetraglow readout or the LDH readout. This next slide demonstrates how you can inhibit glutamate toxicity in eye-cell glutoneurons by application of AP5 and DNQX. We evaluated cell death of eye-cell glutoneurons across 3 different lots when treated with either glutamate alone or in the presence of AP5 and DNQX. And you can see in all 3 lots, when AP5 and DNQX were applied, that the cell death curves were right-shifted. Across the 3 lots, the EC50 for cell death without the inhibitors was consistent, and with the inhibitors, the right shift of the kill curve was also consistent for EC50s. Now, the last part of my talk is going to focus on looking at activity of eye-cell glutoneurons on the multi-electrode array platform, and we focus our activities here on the Axion Maestro 48-well plate system, and we think that's a very nice platform for studying neuronal toxicity when looking at synaptic activity. This graph basically shows the activity that you see when you apply eye-cell glutoneurons to the MEA system. The top raster plots show that each individual tick shows when an action potential happens across the 16 electrodes shown in each plate. There are 3 conditions that are shown in this particular slide: eye-cell glutoneurons mixed with a small 20% contribution of our eye-cell neurons, as well as eye-cell neurons alone. Focusing on the first condition, isolgluteneurons, you can see that the cells have a big basal background activity that approximately every 50 seconds you see a large burst of synaptic activity showing that the cells are communicating and they will fire together. Now, when you mix in a small percentage of isol neurons, which adds an inhibitory component to the cell culture, you can see an increase in the frequency of the bursting events and a slight decrease in the mean firing rate and the frequency of the firing across the activity, either both in the bursts and in the background as well. When you look at isol neurons alone by themselves, which is in the third condition there, you see no synchronous activity and minimal firing on their own. So this really demonstrates a couple important points. The isolgluteneurons have spontaneous synchronous activity, and this activity can typically be seen within a period of 2 to 3 weeks when the cells are cultured on the MEA system. This activity can be modulated by the treatment or the addition of other cellular components, such as inhibitory neurons, in the case of isol neurons as well. And we think this really, when you have both populations, isol neurons and isolgluteneurons, you can really customize the conditions that you want to study in the dish. And importantly, how does this synaptic activity that's exhibited by isolgluteneurons in the MEA, is it amenable, or do you observe normal biological phenomena such as being able to inhibit the synaptic activity, again, with treatment with AP5 and D and QX? That's demonstrated on the data in this slide, where we took isolgluteneurons cultured on the MEA, and we first measured their activity, and you see spontaneous bursting activity at the time 0 there, either in the control wells or the cells not yet treated with AP5 and D and QX. The next condition is what the cells look like 45 minutes after addition of the inhibitors, and you see in the treatment conditions complete removal or no observation of the bursting activity in the isolgluteneurons with AP5 and D and QX. In the controls, the vehicle alone shows no perturbation of the bursting activity. And then when you wash out the AP5 and D and QX and measure the bursting activity just a short while later, after several minutes, about 25, 35 minutes later, you see the cells that were treated with the inhibitor but had it washed out resume their synaptic activity. And again, the control wells are bursting as they would normally with no treatment. And that's quantified in the charts below, really demonstrating in a quantitative fashion that AP5 and D and QX can block synaptic activity in isolgluteneurons, and this inhibition is reversible. So in summary, our isolgluteneurons are a glutamatergic-enriched population of human cortical neurons. They can be used for studying glutamate excitotoxicity using a number of different assay readouts, including cell titer glow or lactate dehydrogenase accumulation. Within a few weeks of post-thaw culture, these isolgluteneurons form robust synaptic connections that can be quantified using multi-electrode array systems. And the isolgluteneurons respond appropriately to known inhibitors of synaptic activity. With that, I will conclude my talk. Thank you.
Thanks, Brad. That was a great presentation to get us started. You provided us with some nice insight into the benefits and capabilities of the isolgluteneurons for glutamine excitotoxicity studies. Before we move on to Andrew's presentation, I'd like to remind everyone again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. So, Andrew, the floor is now yours.
Hello, everyone. Thanks for joining us today. My part of the webinar will focus on a pharmacological case study using the cardiac glycoside Wathan, which is a unique compound that highlights the value of a comprehensive approach to in vitro cardiac safety testing as advocated by the CIPA initiative. In cardiac safety pharmacology, the current strategy for de-risking drugs for torsade de pointe is based on ICH, S7A, and S7B guidances, as well as E14 guidance, and is really focused on evaluation of two surrogates of torsades, QT interval prolongation and inhibition of the hERG potassium channel that is responsible for the rapid phase of repolarization in the heart. So the current derisking strategy has proven to be highly successful. So the table shows a list of drugs that were withdrawn from the market due to cardiac risk. Starting at the top in 1998, terfenadine was withdrawn from the market. It was the first non-sedating antihistamine marketed as Seldane in the United States. And it was a great drug, actually. It was effective. I took it as a kid. But in a small percentage of patients, typically when antifungals, CYP inhibitors were co-administered, there was incidence of torsades, and the drug was withdrawn. You can see a regular pattern of withdrawal up through 2003 when levomethodil was withdrawn from the market. And then after that, up till the present, 2016, so 13 years later, there have been no drugs withdrawn from the market due to cardiac risk. And so the strategy of looking at hERG channel inhibition together with QT prolongation and assessing risk has been very successful in removing, from preventing advancement of dangerous drugs to the market. With the advent of new techniques, availability of cardiomyocytes derived from induced pluripotent stem cells and computer modeling, the current strategy is being re-examined, not just to identify with better sensitivity and selectivity, specificity drugs that have risk, but also to allow advancement of drugs that maybe would be flagged as dangerous due to hERG block, for example, and be removed from drug development, but also to get a more comprehensive view in the in vitro stage of testing of drug development. So this comprehensive in vitro polyrhythmia assay, or SIPA as it's referred to as, is really looking at, as I mentioned, functional effects on not just the potassium channel, which is shown in the top left here, but also sodium and calcium channels. These are all heterologously expressed in HEK or CHO cells, for example, but also in silico models and electrophysiology of cardiomyocytes derived from induced pluripotent stem cells. And together, we can get a better in vitro polyrhythmia risk assessment, or that's the goal of the SIPA initiative. So today I'm going to focus on a single compound, Wathan, because it's a nice case study, and I think it illustrates the advantage of using a more comprehensive in vitro approach during drug risk assessment. So Wathan is a natural compound. It's plant-derived, and it's been used for centuries. It historically was used in Africa in warfare and hunting. It's a cardiac poison at high levels, so people would coat arrows with it, and when it got into the bloodstream at high levels, it would induce cardiac arrest, so pretty effective. It's now known to be a cardiac glycoside, and it's a positive inotrope. The chemical structure is shown on the slide as well. So what is the mechanism of action, or MOA? Well, the first step is we looked at ion channels, not just the hERG ion channel, but also sodium 1.5, which is the cardiac sodium channel, KVL-QT-Min-K, which is responsible for IKS, KIR 2.1, which sets the resting potential, and the L-type calcium channel, CAV 1.2. And you can see for both Wathan and digoxin, which is another cardiac glycoside, there's really no meaningful inhibition at relevant concentrations for either drug on any of the channels. So the mechanism of action of Wathan is not via a direct effect, acute effect on cardiac ion channels. So what is it doing? What is the mechanism? So we went back to the toolbox and we looked at in vitro impedance and field potential signals that were measured from the I-cell squared cardiomyocytes, and we used the ACEA cardio ECR instrument. This instrument is very nice. It has both impedance electrodes and field potential electrodes in a single well. So these cardiomyocytes are plated on top of the electrodes, and we can measure, as you see on the top, the field potential, which is an ECG-like signal. At the same time, we measure the impedance, which is really measuring the movement of the cells on top of the electrodes, and we call it the impedance twitch. You see these positive deflections in the ohms, which is the impedance signal. This slide is just meant to show before and after application of Wathan in black and red. You can see the field potential and the impedance signals. On the left is kind of a longer time scale. On the right is just measuring the initial part of the signal. You can see on the top that there's a deflection in the field potential, but in this initial phase, but not much of a change in the impedance signal, which is as we expect since the cells fire and then contract. So one of the major effects of Wathan is a shortening of the field potential duration, which is the QT-like signal. You can see in the control in black that the FPD is maintained relatively flat, whereas in the presence of Wathan at the various doses, we get a shortening of the field potential duration over time and the concentration. Again, looking at the field potential signal, and this slide is just really meant to show these are exemplar electrodes for the vehicle, and then 2.5 and 10 nanomolar Wathan. You can see over concentration and time again, there's a slowing of the beat rate, there's a prolongation of the field potential duration, and eventually we reach a kind of fibrillating state, which looks like a flat line in these traces at 10 nanomolar. You can look at 3 and 4 hours, for example. And then we plotted here dofetilide, which is a pure hERG channel blocker. At 100 nanomolar, it induces arrhythmias, but by a different mechanism. We know the mechanism to be a block of the hERG channel. So both of these drugs, both Wathan and dofetilide, are arrhythmogenic, again, showing the field potential and impedance signals. You can see that the ultimate signal is different, and it's probably related to the two different mechanisms of action. These plots are a little bit complicated, but they're very, very descriptive. These are plots we make using macros we built in-house. So we export the impedance signal from the instrument, and then we take averages within a well. So over a 60-second recording period, we overlay and average all of the signals, and then we average across wells. So we get a grand average or an average of averages. And that's what's plotted in these plots. So in black, we have the baseline, and then after exposure to the drug, that's the red traces. And they're plotted as means plus or minus 95% confidence intervals. So anytime you see white space between red and black, that means there's a statistically significant difference in the signals. So across the top or the x-axis, we have an increase in concentration of Wathan, and then going down the y-axis, the increase in time, so out to 4 hours. And the point of the plot is that you can see a clear concentration and time dependence to the effect. The other thing to point out here is I drew a blue box around the concentrations and time points that showed arrhythmias, and the arrhythmias manifest themselves in this type of analysis as these kind of angular shapes at the end of the signal. That's just the way the software works. But anytime you see these, that means there's arrhythmias in the signal. Now, interestingly, if you record out to 16 hours, and one nice thing about these cells in this system is you have the plates in an incubator, you dose the plates, and you walk away. And you leave them in the incubator as long as you would like. In this case, we went out to 16 hours. You can record longer, but it's label-free and non-disruptive. So really, we've recorded out to even up to a week or more. And what was interesting here is that 16 hours there was a left shift. So previously at 5 nanomolar we didn't see any arrhythmias, and now you can see, at least at the 8, 12, and 16-hour time points, that there are arrhythmias in the signal. So we were curious about this. We know that there's no direct block of the hERG channel or the other major cardiac ion channels. So we wanted to do a longer-term functional study, and the way we did that is shown in the next slide. So it's a little bit busy, but I will explain it. The way this is done, it's using the Ironworks Barracuda system and the hERG cell line that we have in-house. And we expose the cells to whatever compound we would like or vehicle control out to 24 hours. And then we measure density of the current in the cell membrane. And after 1 hour, you can see that there's really no effects in the top right of digoxin or Wathan. But after 24 hours, there's a clear concentration-dependent effect seen with both of these cardiac glycosides, also in the positive control, which is pentamidine, a known hERG ion channel trafficking inhibitor, but not in the vehicle control. And on the bottom, you can see the dose response curves for the two drugs. So we think that in addition to a non-ion channel related mechanism, there may be an ion channel trafficking issue with a hERG channel that contributes to an increase in the potency of the Wathan effect that we see in the cardiomyocytes over time. So in conclusion, what we've observed is that there are no direct effects on the cardiac ion channels, but there are clear effects on the electrical field potential and impedance signal of iPS-derived cardiomyocytes. This is not very surprising. It's known that Wathan inhibits the sodium potassium ATPase, which then indirectly affects the sodium-calcium exchanger, altering calcium homeostasis, and it's believed that that's the mechanism of action. If you were to screen using only cloned ion channels, you would not pick up this liability. So we think this kind of speaks to the power of the cardiomyocyte addition to the in vitro screen. The table shows, in the summary and conclusion, shows that the cardiomyocyte results, and they do match the in vivo results. We published this at SOT in 2016, CRL did, but there's a shortening of QTC, increase in the RR interval, and arrhythmias, their time and dose-dependent change is seen in the canine model. So all of this is consistent. Finally, the final point to make is that the trafficking effect may be an important consideration when looking at a thorough in vitro evaluation of potential cardiac liability of drugs. Pentamidine is a classic example of that. No short-term hERG effects, but there is a longer-term effect. And Wathan may be showing a similar, or maybe even the other cardiac glycosides may have similar type trafficking effects. Thank you. That concludes my part of the talk.
That was great. Thanks very much, Andrew. You described for us a great use of the I-cell cardiomyocytes and their benefit to detecting cardiac risk compounds. It looks like we're getting some really great questions coming in already, but I'm encouraging everyone in the audience to keep them coming. Simply type your question into the Q&A box on the left-hand side of your screen, and hit submit. So let's get our final presentation of the webinar underway. Will, you have our attention.
Thank you very much. The title of my talk today is Evaluation of icell hepatocytes 2.0 in two-dimensional and three-dimensional cultures for preclinical toxicity testing. So hepatotoxicity is one of the major sources of preclinical and clinical attrition as well as post-market withdrawal of therapeutics. So the main question we always ask is, why do we fail at identifying human hepatotoxicants early in drug discovery and development? And there's three main reasons for this. The preclinical in vivo studies have very poor concordance with identifying human hepatotoxicants, and that drug-induced liver injury is often considered idiosyncratic, but there's a lot of host factors and intrinsic molecular properties that are often implicated retrospectively. And drug-induced liver injury is often multifactorial and difficult to predict in vitro with any one particular assay. And if we look retrospectively at all the epidemiological factors that are driving drug-induced liver injury over the past 50 years of studying this, we kind of have three buckets of properties that characterize risk for drug-induced liver injury. In particular, the physical chemical properties, how lipophilic and polar the molecule is, as well as the charge can drive certain features of drug-induced liver injury. And then we know a lot about the structural alerts, in particular metabolic alerts, that would lead to reactive metabolite formation. The disposition of the molecule also holds a lot of risk for drug-induced liver injury. How high the dose and the total exposure, as well as if it's highly metabolically cleared, if it's predominantly excreted in the bile, and whether there's reactive metabolites formed, and then more recently, whether it inhibits different transporters responsible for bile acid homeostasis. And what we're going to discuss today is really there's also in vitro cytotoxicity and toxicity assays that have been shown to be quite predictive retrospectively of identifying human hepatotoxicants. So at Genentech and at many other companies, we employ a battery of different assays and parameters to assess risk for drug-induced liver injury. And that includes mitochondrial toxicity assays, transport inhibition studies, even considerations for in silico tools and physical chemical properties, as well as DMPK-related parameters such as metabolic stability and the presence of glutathione or cyanide trapping. Today, we're really going to focus on looking at different models for cytotoxicity. The gold standard for assessing hepatotoxicity and overall cytotoxicity is really using primary human hepatocytes. And this is scalable for up to 384 well or more, has relatively good positive predictive value, and depending on what endpoints and studies have been done, it can range between 50 and 90%. It's amenable to high throughput screening, and it's capable of detecting metabolic-driven toxicities. The real disadvantages of using plated primary human hepatocytes is you have poor negative predictive value. It's basically a coin flip if you have no cytotoxicity, whether or not you have a bad actor or not. And then you have rapid loss of liver phenotype during culture. And you can really only measure acute cytotoxicity, and it's not amenable to repeat dose. And that's due to the fact that it basically, the cells lose P450 activity and liver phenotype within about 48 to 72 hours. And there's also lots of variability in performance that make this a little bit more difficult to employ over long periods of time. So we became interested in exploring iPSC-derived hepatocytes to look at another model for addressing cytotoxicity as a risk for hepatotoxicity. And what we're showing here is the icell hepatocytes 2.0 and some morphology and hepatocyte markers. And that the panel on the left top corner just represents phase contrast, where you can see the very unique hepatocyte-like morphology of the kind of honeycomb structures, binucleation, and forms of tight junction or canicular membranes. Early on, after plating, we can actually see pretty robust expression of the hepatocyte nuclear transcription factor, HNF4 alpha. And then over time, you see albumin production in these cells, and also about 7 to 10 days, we can see actually occludin expression outlining the canicular membrane tight junctions. So all in all, these look very hepatic-like and are quite promising. The advantages that we see with using the iCell Hepatocytes 2.0 is that you don't need to overlay with Matrigel, which was the previous version had to do. And this makes this a little bit more easy for us to employ in our normal practices and transferable to our primary human hepatocyte assay. The advantage that we're really looking at for the iCell Hepatocytes 2.0 is not to replace our primary human hepatocyte acute cytotoxicity assay, but to really look at ways that we can use this model to assess long-term cytotoxicity or repeat dose cytotoxicity. And this is just some phase images of the iCell Hepatocyte 2.0 in 7-day, 14-day, and even up to 28 days in culture. Now, CDI has done some characterization of these cells and knows that P450 expression is pretty robust all the way through day 10, and that at least albumin production is consistent through a month. And so what we wanted to do is really assess, could we use iCell Hepatocytes 2.0 to look at long-term exposure? And so what we set up was a study to assess a 2-day or 48-hour drug treatment, as well as a 7-day exposure with 3 doses. And the first thing we needed to do to determine if this was feasible is to assess vehicle tolerance. And so what we're showing here in the plots are the cell viability as a function of DMSO concentrations, either in 48 hours or 7 days. And in this study, in the box that are highlighted is at 0.5% DMSO, we don't see a significant drop in overall cell viability, either at 48 hours or in 7 days exposure. And this is promising, and so we proceeded to set up a proof of concept study to address long-term cytotoxicity in the system. So the way that we set up this study was we basically had 40 commercial available drugs that were evaluated. These are a mixture of known hepatotoxicants and structurally similar controls. And what we did is we plated the iCell hepatocytes and changed the media, and then basically at day 5 we dosed for compounds for 48 hours and took down a set of plates at 7 days. And then we continued to dose even up to day 9, and then took down the assay plate number 2 at day 12, which would have been 7 days of treatment. We did an 8-point IC50 curve starting at 100 times the Cmax or the limit of solubility in the vehicle of 0.5% DMSO. We looked then at cell viability using the cell titer glow assay, which looks at cellular ATP and a luminescence readout. And so what we're going to show is just some of the preliminary data that we have from this 40 compound set, just to kind of show you the differences that you see when you culture the same cells either in 2 days or in 7 days. So looking in the left panel here, we have acetaminophen, which is kind of the gold standard for metabolic-driven cytotoxicity. And you can see at 2 days we see absolutely no cytotoxicity in the iCell hepatocytes up to about 10 millimolar or 6 millimolar. I think it's maybe 5 millimolar. And then at 7 days, you can see significant improvement in the readout there. Similar, ketoconazole, we see a leftward shift of about 3 to 4-fold. And then for mitomycin C, you see a significant leftward shift. And this is to be expected for most compounds. We wouldn't expect to see a rightward shift in these assays. And then we also looked at some controls here. And we looked at ambrisentan, which showed no toxicity in both 2-day and 7-day. And then looking at diclofenac, which is also metabolically driven cytotoxicity, it forms a quinonamine as well as acylglucuronides. And we can see that only at the high dose do we see any cytotoxicity, but it does give you a signal that you didn't pick up at 2 days. And then for nifazodone, we don't see much of a shift at all, although the curve shape and hill slope is a little bit better at 7 days relative to 2 days. So all in all, this was quite promising as a preliminary look, and we're continuing to evaluate the 40 compound set and to determine what the increase in sensitivity and specificity to identify hepatotoxicants in this small test set. since we had all this data, what we really wanted to look at is it's well understood that iPSC-derived hepatocytes in a two-dimensional culture, although they do demonstrate robust liver phenotype and metabolic capacity, they still underperform relative to the metabolic activity of either suspended or immediately plated primary human hepatocytes. And so what CDI has been working on that we're very interested in is really taking the iCell hepatocyte 2.0 and plating them and then doing a gentle cell disruption to take them off the two-dimensional configuration, and then they seed them in round bottom, low binding plates, and they form spheroid cultures. And this has been a hot topic in drug discovery and toxicity testing for the past 5 years or so, as we know that in a 3D configuration, most cells tend to behave a little bit better and more physiologically relevant. And so what we're going to show just now is some preliminary data using the iCell hepatocyte 2.0 in spheroid cultures. And the first thing that they did to characterize this, which is quite promising, is just looking at one cytochrome P450 3A4 activity in two-dimensional iCell hepatocyte 2.0 cultures or three-dimensional spheroids. And what you can see is that immediately in spheroid culture, you see a 5-fold increase in 3A4 activity versus the two-dimensional configuration of the same cells. And this is consistent with what is known with primary human hepatocytes when you put them in three-dimensional culture. So this is quite promising, and we're really excited about further characterizing these spheroid cultures to enhance not only the metabolic capacity, but potentially the physiologically relevance of these models, and maybe even more adult phenotype as they are cultured. And so when you look at the spheroids, and we're just looking at the uncoupling agent FCCP here, which you can get IC50s whether you plate 2,000 cells per spheroid, 1,000 cells per spheroid, or 500 cells per spheroid. And what we're seeing here is just the IC50s of 7, 4, and 4 for all three of these preparations. But the advantage of looking at the spheroid cultures is that even with 500 cells per spheroid per well, you can actually get very nice signal to noise with the cell titer glow assay, and you get very reproducible IC50s, indicating that you can miniaturize this application to use for drug discovery and in turn reduce the amount of reagents and cells that you need to do screening. So this is quite promising, and we're looking forward to employing this here at Genentech to see how this works. And so here's just some preliminary comparisons of the two-dimensional cytotoxicity of diclofenac, mitomycin C, and tolcapone in two-dimensional 2-day and 7-day cultures of iCell hepatocytes 2.0 versus a 7-day culture in the spheroid cultures in three-dimensional. And what you can see is that for diclofenac, although the curve in the hill slope is quite steep, we see a significant drop, at least at the highest concentration. So not very huge differences, but promising in the right direction. For mitomycin C, this kind of flips the profile from what we expected to see. And this is very unusual when we're trying to understand why this behavior in three-dimensional is significantly different than in the two-dimensional plated configuration. But with tolcapone, you can see a nice leftward shift versus the 7-day two-dimensional cultures versus even the 2-day two-dimensional culture. So this is quite promising, and we're really looking forward to exploring many more compounds, including our own proprietary compounds in these assays. So to conclude the talk that I did is basically iCell hepatocytes are capable of both short and long-term treatment while maintaining their liver phenotype, and I think moving towards a three-dimensional configuration will really enhance their physiologic relevance and also their metabolic activity, and likely increase them to more of an adult phenotype and more comparable to primary human hepatocytes. And that the 3D culture enables us to really kind of miniaturize this to a point where we can use a lot of wells per vial of iCell hepatocyte 2.0, which is nice for scaling this up for screening applications. And that further studies are underway to continue to evaluate the overall predictive value for our identifying human hepatotoxicants in both two-dimensional and three-dimensional long-term cultures. And the other considerations that we're really interested in is really using this type of model to also further explore target-related effects and other gene perturbations, and using their micelle approaches of doing CRISPR-Cas9 deletion or insertion of genes. Having a robust I-cell hepatocyte model to test these factors is quite interesting and beneficial for drug discovery and toxicity testing. Thank you.
That was excellent, Will. Our audience now has a greater understanding of the versatility of I-cell hepatocytes 2.0 and how they can be used in 3D culture systems for high-throughput screening assays. Before we start the Q&A session, I want to let everyone know this is your final chance to submit your questions for our speakers. So hurry up and send them in now. All right, it looks like we have a bunch of good questions already, so let's begin the Q&A. And we'll try to get to as many questions as possible. All right, everybody, so we'll start the Q&A session. Our first question is going to be for what would be Brad, but Chris Kannemeyer is online. He's a group leader at CDI, and he'll be answering the questions for Brad today. So, Christian, the first question we have from one of our audience members says, I understand what neurons are, but what exactly are gluteneurons?
Yeah, hi. So this is Christian Kannemeyer. So our gluteneurons are cortical neurons that are excitatory as opposed to our product, the I-cell neurons. So we really developed the gluteneurons as an additional product for the I-cell neurons, which are excitatory, and we were really looking to develop this product to show the macro-synchronous network activity. And so because they're glutamatergic and they're excitatory, we call them gluteneurons.
Okay, great. Thanks very much. Andrew, we have a question for you. Our audience member wants to know, do you have other examples where cardiomyocyte physiology correlates with in vivo data?
Thanks for the question. Yes, we had a presentation at SOT this year, and we showed using the same platform and the I-cell squared canine heart failure drug example. And so at CRL we have here in Cleveland we have our in vitro ion channel and cardiomyocyte capabilities, and of course we also have the in vivo facilities. And so we did a in vitro to in vivo comparison of pimobendan, which increases contractility in the canine heart failure model. And what we showed is the impedance twitch parameters were modified in expected ways upon exposure to the drug and correlated with the in vivo canine findings. So that's one example.
Okay, great. Thanks, Andrew. Will, we have a question for you. Someone in the audience would like to know, do the cells demonstrate long-term function over the duration of compound treatment?
Thank you. It's a great question. And so we chose for the studies we presented here and a lot of the data that we're not showing here, 7-day treatment, which we characterize stable P450 activity, at least in albumin production over that time. And so we didn't extend this to, say, 14 days, which some of the phenotypic markers may be changing. So we did try to pick the maximal time point that we felt pretty comfortable was going to distinguish 2 days from 7 days, and that we would basically have a very stable model in that time frame.
Great. Thanks very much, Will. Christian, another question for you. Question goes, bear with me, this is a bit of a tongue twister. Besides glutamic acid, have you looked at cytokinic acid effects in the cytotoxic assays with iPSC glutamatergic neurons, and do the cells respond in a similar fashion to glutamic acid?
So yeah, thanks for the question. So we have looked at excitotoxicity on our glutaneurons with glutamic acid and the data you have seen, but we have also tried canic acid. And it works as expected. So you also see an effect on our glutaneurons from canic acid, which I think is expected from the biology.
Okay, great. Thanks, Christian. Andrew? Question for you. Has iPSC toxicity testing been accepted by the FDA as an alternative to in vivo testing?
Hi. Yeah, so the answer to that, I think, is not yet. And I mentioned in the talk, and as probably most people are aware, the ongoing efforts to refine in vitro testing. These are ongoing and fairly large efforts involving members of industry, pharmaceutical companies and federal regulators. And so the cardiomyocyte part of that, the pilot study is done, and the larger study is going to start soon. So that's kind of the long answer to the question.
All right. Thanks very much, Andrew. Will, another question for you. One of our audience members asked, do you expect differences in sensitivity in 2D versus 3D hepatocyte culture?
Great question. We've done a lot of work with primary plated hepatocyte or primary human hepatocytes in spheroid cultures versus plated configurations, and we do see a pretty significant enhancement of sensitivity to a lot of clinically relevant hepatotoxicants. And so this preliminary study with the CDI spheroids versus two-dimensional culture is looking promising in that same direction, and there's always going to be some compounds that, like I showed here with mitomycin C, that seems to basically buck that trend. But for the most part, we are expecting enhanced sensitivity, and I think as CDI and we further characterize the spheroid cultures, I think it's getting to be more widely accepted that when you do put cells in a three-dimensional configuration and you remove the plastic contact and you have this more physiologically relevant configuration, that you do enhance P450 activity, which we showed here, as well as other predominant liver phenotypes that we expect to enable us to pick up more signals than we would in a plated configuration.
Great. Thanks, Will. Christian, another question for you. Are the I-glutamate neurons coming from neural precursor? How is the purity being evaluated if that's the case?
So our process starts with induced pluripotent stem cells. So we have an induced pluripotent stem cell population that we then guide to be differentiated into excitatory glutamatergic neurons. So they, as per biology, they have to go through a precursor stage. The purity that we are looking at these is that we know that the cells at the end of the process are MAP2 positive, they're TOUCH positive, they're also nestin negative. And on top of that, we also employ our single-cell gene expression that we have shown in the presentation, which basically tells us how the population is made up in terms of glutamatergic and GABAergic neurons. So the answer to that question really is that we are not starting from a neuro precursor, we're starting from iPS cells, but the differentiation process has per biology go through a neuro precursor stage.
Okay, great. Thanks very much. Andrew, another question for you. Our audience member says that they're not sure they understand why the decrease of the hERG signal by Wathan takes 24 hours to show.
Hi, yeah, thanks. That's a great question. So we showed in the early part of the talk the acute studies where there really isn't any block of hERG current under short-term acute administration. So application of, say, 5 minutes, perfusing the cells or exposing the cells. And then in longer-term functional studies, that was at the end of the talk, there was a change in the impedance signal and also in the hERG current levels. And that was done, that was a functional assay using the Ironworks Barracuda and our clone cell line. And so we don't know for sure the mechanism, but the time course suggests that there's something with insertion of the channels into the membrane or trafficking to the membrane that is underlying the longer time course for that.
Oh, great. Thanks very much. Will, we have another question for you. Audience member says that you mentioned that you were not looking to replace primary hepatocytes in your assay, and he would like to know why not.
Great question. I think, maybe I should qualify that question a little bit more. I think today we are not looking to potentially replace primary human hepatocytes based on using, say, iPSC-derived cells or other liver platforms as a first-tier screen, simply based on the fact that we have extensive historical data with this particular model. And we do a pretty good job of characterizing a lot of hepatocytes that we use. And I think we understand the predictive value, although, as I stated in the talk here, it's kind of a coin flip if it's negative, whether you really have a cleaner compound or not. But it's kind of the gold standard today. And until we can, run hundreds and hundreds of compounds through different systems to basically show superiority to what is our gold standard, it's going to be kind of fixed in our in vitro screening paradigm at this point. I think what we're really looking for is kind of that second-tier assays that give us, a little bit enhanced sensitivity. So if something comes up relatively clean in the primary human hepatocyte assay, that we can pressure test it either by duration or more physiologically relevant models that would give us, more sensitivity to pick up signals.
Great, thanks very much. Christian, back to you. How frequently do you need to change the media when culturing gluteneurons?
So for our gluteneuron culture, when we have them on the MEA, on the Axion MEA, we change medium every other day. And but just a word for medium, we know that it is absolutely essential to use the BrainFizz medium, which is just came out from Stem Cell Tech. And that is absolutely important to remind everybody is that the medium that we use is absolutely essential to see the proper functionality of these cells.
All right, thank you, Christian. Andrew, question for you. Are you defining hERG trafficking based on functional current block or channel expression?
Yeah, again, thanks for the good question. So we have two different assays that we run here. One we call hERG Light or Chan Light, and that's a labeling assay, so we have a tagged channel. And then we use luminescence and fluorescence after the channel is tagged to look at channel levels in the membrane. And but what I showed today was a functional assay that looked at current levels, actual current flowing through the hERG channel before and after exposure to the drugs. And that was a 24-hour exposure. So we incubate the cells overnight, basically, and then look at the levels. And we're able to do that at sufficient numbers due to the platform that we use. It's not manual patch clamp. It's the Ironworks Barracuda plate-based assay.
Great, thanks, Andrew. Will, question for you. Have you considered the impact of other liver cell types on the modeling of hepatotoxicity?
Thanks. Great question. This is kind of an area that, also has been, pretty much widely studied, and it's in continuing growth in terms of what heterotypic and homotypic interactions that you need to recapitulate a liver in a dish. Some of the things that we're interested in, just to get back to CDI's eye cells, is they have a macrophage model or monocyte model that, once we can kind of iron out some of the media consistencies between the two, it would be really interesting to take their eye cell hepatocytes and their monocytes and do a co-culture in the typical 10-to-1 or 6-to-1 ratio that people have done with primary non-parenchymal cells and hepatocytes. And we know, not just from an inflammatory standpoint, but having multiple cell types that represent the liver lobule and the different cell types represented in vivo do enhance the performance of these models, at least for subsets of compounds that have been thrown at them. So we're excited about looking at that for this particular model, as well as we extensively are looking at that in other hepatic models as well. But great question. I think it's something that we'll find out a lot more as more and more data is generated.
Great, thanks, Will. Looks like we have one last question for Christian. Christian, for the I-glutamate neurons, are you using glia to co-culture them on the MEAs? Does that make a difference versus culturing the neurons alone?
Well, that, of course, is also a great question, and I think everybody's really interested to find out the answers. But at this point, the experiments are ongoing, and we're currently doing these experiments, so I'm not quite there yet to provide an answer. Okay, great. Thanks. I think we have time for one last question. Looks like it's going to be for Will. Will, in tox testing with eye-cell hepatocytes, 2D or 3D, what endpoints are measured?
So, great question. For this particular study, we actually looked at three endpoints, but what we presented is really just cell titer glow. This is the total cellular ATP. So we're looking at a decrease in cellular ATP that would be a surrogate for viability. We also looked at live cell staining and also dead cell staining at the same time. So we multiplex this, and just for the simplicity of this particular presentation, we're just showing the cell titer glow assay. But I think you can use a lot of different endpoints, and whether it's LDH release, if you can get a consistent readout with that. But most, pretty much what we are most comfortable with is really looking at ATP readouts or some other just general cytotoxicity assessment.
Great. Thanks very much, Will. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site for 6 months at www.genengnews.com. So if you missed any part of it or want to watch it again or forward it to your friends and colleagues, which we always encourage, that would be great. I'd like to thank Brad, Andrew, Will, and Christian, again, for their informative presentations and answers. And I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to CDI Technologies for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.